Okay. So the test material begins with what was numbered as sort of handout number 3. The first test, remember, was handouts 1 and 2. And 2 was about B cells and antibodies. And this one here was the next one that came along. And we're going to come about T cell receptors, T cells and T cell receptors. And this set of notes also includes discussion about MHC molecules and residues and all that stuff. So we began by pointing out the evolutionary relationship, as we were just talking about this afternoon, between the T cell receptor and one of the arms of an antibody molecule, right? So the beta chain resembles part of the heavy chain, okay? And the alpha chain resembles the so-called light chain. And then we looked at the crystallographic structure of the T cell receptor, and the alpha and beta chains each have 3 hypervariable loops, all very analogous to the arms of an antibody molecule. These are the hypervariable things. This is where the VDJ stuff goes on in here. Okay, and this, the whole surface here lines the peptide, foreign peptide plus the MHC molecule. That's what we're saying. Okay, and we said that like B cell development, you create a gene for an antibody molecule by assembling little pieces of a gene, little segments, bringing them together to form actual contiguous nucleotides to function as a gene for the T cell receptor proteins alpha and beta. And we said that there's a little semantic thing here. We talk about the T cell receptor. Often we mean just the alpha beta guys here. But in other contexts, when they say the T cell receptor, they mean this whole complex here. We might try to distinguish by calling the T cell receptor complex here, where we have the alpha beta chains plus the so-called CD3 antigen, or the CD3 proteins, or the CD3 complex here. These have little Greek names, gamma, delta, epsilon, zeta, and so forth. The cytosolic tails of these proteins initiate signal transduction events, so that when the T cell receptor recognizes its contact end, its cognate antigen, we will then activate these CD3 complex proteins and also the zeta chain to initiate signaling. We'll have a lecture where we'll be talking about the signal transduction events and so on. And then again, as you all know, we have, in addition to the classic alpha beta, we have some cells which are gamma delta T cells. And we'll get into more detail about all this afternoon. And then, of course, I'm telling you for the first time then that T cell receptors on a T cell, they see their cognate, this is on page 60, they see their cognate antigen as something that's a peptide, a piece of a protein, a peptide that's being presented to the T cell receptor by an MHC molecule. Okay. And we said that there are 2 kinds of MHC molecules. Class 2, which have 2 transmembrane segments, and class 1, which have just the 1 transmembrane segment here. I'm sorry, class 1 up here, and class 2. Okay. And that the CD8 protein, its function is to bind to the base of an MHC molecule. Okay. Therefore, give it the type, contribute to the overall tightness of the complex. Also, just as the CD3 complex has binding sites for signal transduction molecules, the CD8 protein also have this Stark family tyrosine kinase that's associated with it. And the same thing for the CD4. So the CD4 and CD8 have 2 functions. One is to bind the base of the MHC, therefore tightening this whole complex, but also that contributing to the signal transduction events. And we talked about the structure of an MHC1 molecule, for example, here. It has a little beta sheet platform, and along the lengths of the, if you look at it from above, for both class 1 and class 2, we have a beta sheet floor, and then those little mountain walls here are alpha helical structures here. And the peptide sits on the valley floor and between the alpha helical mountains. And class 1 molecules, the end of the valley is closed, so the peptide has got to be within the valley. For class 2, the end of the valley is open, and we can have extra amino acid sequences draped over on either side. So the class 1 molecules are typically 8, a minimum of 8, sometimes as many as 10 amino acids displayed on this floor. In class 2, you can get from a dozen to 2 dozen amino acid long peptides bound. And just to remind you, we talked about the fact that an interesting feature of these MHC molecules is that they can bind and present, they can tightly bind and present peptides, thousands of different peptides. Okay. So how can you be able to present thousands of different amino acid sequence peptides, yet they're all very tightly bound? Okay. And part of the answer we said was that all peptides have a positively charged amino group at the amino end and a negatively charged carboxyl group at the other end. So all peptides have that in common, so all peptides can be bound based in part just on that. Okay? But then to get sufficiently tight binding, you need to have, for each MHC1 allylic version, okay, you're going to recognize certain features of the peptide. For example, all the peptides they're going to bind to an MHC1 protein allele number, it's MHC1A gene, allele number 17, okay? 10,000 peptides can bind to that, but all 10,000 peptides all must have, for example, a tyrosine or phenylalanine here at this position, and then the carboxyl terminal end will have not just the fact that it's negatively charged, but it must be leucine in this case, okay? And these amino acids, this tyrosine and phenylalanine, the side chains on these two are very similar. They have basically a benzene ring. For phenylalanine, the side chain is just a benzene ring. For tyrosine, it's a benzene ring with a hydroxyl group on it. So these have structural similar features here, and they can fit into a slot. So here's the MHC molecule, here's the peptide. The phenylalanine slips into a little slot here on the floor of the valley where you get very tight binding. Okay? So these are the so-called anchor residues, which anchor the peptide onto the floor of the valley by complementary lock and key type of structures. Okay? So I'm going to ask you a question now. I'm going to show you this. I'm going to say, what does it mean that in let's say this peptide here, you always have tyrosines at position number 2, and the valine or an isoleucine is looking very much like that down here. You're going to say, oh, well, those are the anchor residues. The side chains are very similar to each other, and they fit into tight complementary holes on the surface of the MHC molecule. These give you the really tight binding. Okay. And then we're going to talk about the fact, you're going to remember, of course, that all God's children express MHC1 molecules. All the cells in your body express 10, 20,000 MHC1 molecules all over their surface. Okay? Those MHC1 molecules can be of any one of 6 different flavors, so to speak, because there are 3 genes, A, B, and C, that code for similar but not identical MHC1 molecules. Plus you've got A, B, and C from dad and A, B, and C from mom. So you've got 6 different possible MHC1 molecules, for example. Okay. And all those MHC1 molecules are presenting in their little peptide binding groove here, a peptide derived from a cellular cytoplasmic, or maybe more properly, we should say cytosolic. Cytosolic is kind of the same as cytoplasmic, but cytoplasmic is everything that's outside the nucleus. The cytosol is all the gray floating soluble proteins here inside the cytoplasm. All these peptides come from the cytosol. Okay? So you're presenting, every cell in your body is constantly presenting a cross-section of the peptides from all the tens of thousands of different kinds of proteins that it's making. Okay? But these are self-peptides derived from self-proteins, and the immune system could care less. They don't recognize it. But if you get a virus infection, now we're making viral proteins in the cytoplasm, and peptides, now viral peptides are being presented together with normal cell peptides by all your class 1 molecules. And the killer T cells see those foreign peptides, and they rain down hell on the infected cell, and they kill it. Okay? So that's why all cells make class 1, because almost all cells can be infected by and support the growth of a virus. Okay? MHC1 molecules are essentially an antivirus mechanism. Okay? But then only a few cells make class 2. These are the so-called professional antigen-presenting cells. Okay? We'll have more to say about them in detail, but right now it's macrophages and dendritic cells and B cells. These special cells, they all express class 1 molecules as well, but in addition to class 1, they also make class 2. And you're going to remember that class 2 molecules are presenting peptides from proteins brought in from the outside world, brought into the cell, cut to pieces, and then loaded onto class 2 molecules. You're going to remember that the peptides that get loaded onto class 1 molecules, they come from cytosolic proteins. Here's the cytoplasm, the cytosol. Here's the endoplasmic reticulum. Okay. This is the interior, the lumen of the endoplasmic reticulum. Here's one of these cellular proteins. Could be any old protein, lactate dehydrogenase. It's lived on its sort of 2-day lifetime. The average protein lives a day or few days in the cell, and then it's degraded and replaced with hormone synthesis. This is somehow detected as being a little battered and beaten up, and so it gets targeted by the ubiquitination machinery. We add a string of ubiquitins onto this protein. The ubiquitins tag it for death. Okay, dead man walking. And because when this ubiquitin chain brushes up against the opening of the proteasome, one of the garbage disposal unit, the garbage disposal, the proteins here on the open surface, they grab that ubiquitin chain, pull it in, unfold the cellular protein, stuff it, unfold it through a channel in the middle of the proteasome. The walls of the proteasome are digestive proteases who slice and dice that protein as it passes through the centered passageway. So whole proteins go in one end and chopped up peptides come out the other end. And then this, you'll remember this, the so-called TAP transporter, TAP. Now there's a TAP1 and a TAP2, but you're just going to remember the TAP transporter associated with antigen processing. This is a transporter protein that doesn't transport sugars or amino acids. It brings across whole peptides, 8, 10 amino acid long, 20 amino acid long peptides. It brings them across the membrane and delivers them into the interior of the endoplasmic reticulum. And once they're in here, they can get loaded onto newly made MHC1 molecules, which will then go by vesicle trafficking onto the cell surface. Okay, and here was the details of this. Here's the proteasome, here's the TAP transporter. We've brought these peptides in here. Here is newly made MHC1 molecule here, and here and here. The various chaperones will help this thing to fold, and then we'll make sure that, and then we'll get anchored to this part of the TAP transporter. So it's now anchored there, and these peptides that are coming in will find their way, and we'll see if they can bind to the peptide binding glue of this particular one of your 12 different MHC1 molecules. Okay. Once the peptide has been loaded on, the protein, the MHC1 protein, finally folds to its final stable shape, and that permits it now to leave the lumen of the ER and go by vesicle trafficking in the usual way to the Golgi compartments, and then again by vesicle trafficking fusing to the cell surface. And now that MHC1 molecule is an integral membrane protein on the cell surface, and the peptide is being displayed to the outside world. Okay. We mentioned that many viruses have ways of sabotaging this process. You don't need to memorize what herpes does and what adenovirus does. And then we talked about the details of the class 2 pathway. Now, how come these peptides, which are being brought from cytosolic proteins, these peptides are being brought into the lumen of the ER? Now, in the lumen of the ER, we have the domain of the MHC1 molecules that binds peptides. But we also have the domain of the class 2 molecules, the peptide binding domain of the class 2 molecules. Why don't these cytosolic peptides get onto class 2 molecules? We just said they don't. We said these class 2 molecules only present peptides from the outside world. How come these intracellular peptides are not grabbed by a class 2 molecule and presented to the outside world? Okay. Well, that's because there's a little protein called the invariant chain protein. Okay. Here's the newly made class 2 molecule. Here's all these cytosolic peptides. They can't jump onto the binding group here because the invariant chain is, they've got the little candy cane here, and the handle of the cane sits on the peptide binding group and prevents cytosolic peptides from being grabbed by class 2 molecules. Okay? So that's how we protect class 2 molecules from being occupied by intracellular peptides. And this binding then provides the same stabilizing role as the peptides would. So the invariant chain here, class 2 complex of proteins, is recognized as fully folded class 2, and it goes by vesicle trafficking out towards other parts of the cell. And these vesicles here acidify a little bit, and that triggers the cleavage of most of the invariant chain, leaving only the handle of the chain and the peptide. You'll remember that that is called the clip peptide. So the clip peptide continues to sit in the groove and stabilize this whole protein. And now we go deeper in, and now endocytosis or phagocytosis is bringing in outside world proteins or whole bacteria. They're being digested by the lysosomes, and now the peptides derived from the outside world proteins are now in the same compartment as the class 2 molecule. And you'll remember, not shown, always present, so not shown until now, there's an MHC1 type kind of protein called HLA-DM. You remember HLA-DM. HLA-DM in this acid environment here is able to bind the class 2 molecule here, make it open up and release the clip peptide, and now bind the extracellular world peptide in its place. I said don't worry about page 74, read exon 74. Okay. I don't think I'm going to give you huge detail here, but again, this is just I talked about this. Here is the structure of the MHC1 protein here, and here is the peptide sitting in the groove. Here's the T cell receptor, the alpha chain and the beta chain, and there are just 2 loops. Okay, the CDR3 loops of the alpha chain and the CDR3 loop of the beta chain. Those loops bind the peptide. The CDR1 and 2 loops of alpha bind MHC, and the CDR1 and 2 loops of the beta chain bind the MHC1. So it's just these 2 loops, one from the alpha chain, one from the beta chain, the CDR3 loops. They are the only ones that are going to give you the specificity of binding that foreign peptide. We'll talk about how superantigens can cause what seems to be a TCR receptor MHC1 peptide. These normally wouldn't be brought together and held like this unless this was the cognate antigen of this TCR, but the superantigens sort of cross-link them together and then thereby activate this T cell mistakenly and falsely. Okay. So these bacterial toxins cause massive activation of all your T cells and not specifically. That creates a cytokine storm. It's very toxic. And this just shows that pretty much all cells make class 1. Antigen presenting cells make class 2. And we talked about, again, the polymorphism of the MHC1 genes. Okay, we have on chromosome number 6, A, B, and C genes that make class 1 proteins. But we have the mother chromosome 6 and the father chromosome 6 for one thing. And the mother and the father chromosome 6, the A, B, and C genes are probably different because there's 250 different mutated allelic variants of the B gene, okay? And there's 70 of the C and 120 of the A. So, any two, male and female, come together to fertilize an egg, make a zygote. That zygote's probably going to have a different A gene from the mother as compared to the father. So that's good because you can present more different peptides. Okay. And the mutations that distinguish these allelic variants, they're all located, they all specify different amino acids in the part where you bind the foreign protein, okay, or any peptide. If they're on the floor of the valley or on the walls. Now, each different MHC allele or isoform has a characteristic set of 10,000 peptides. Don't worry about page 82, but memorize page 83. Okay. Joke. Now, the second of the lecture notes, more details on B cell development. You're going to remember that when you're generating the gene to make an antibody molecule, the first thing you do is to try to make an antibody heavy chain gene. Okay? So you're going to have to remember that pro-B cells are cells that are earlier than pre-B. So pre means just right before the finished product. Pro means earlier time. So in the pro-B cells, we begin the process. We start grabbing a D and pairing it up with a particular J. And after we've made a DJ pair, we move on to a late pro-B, where we grab one of the V's and pair it up with the DJ pair that we made. So that when we succeed, we have made a functioning DNA sequence, a gene, for the heavy chain with a particular BDJ set, right? Okay. So we express that newly made heavy chain on the surface of the B cell, and we pair it up with a pretend light chain. Remember, we have to get to make our own real light chain by DJ pairs. We're going to use a pretend light chain. Okay, that was the V pre B and the lambda 5 to these two genes, encode these two proteins here, which together make a protein, a heterodimer that closely resembles the structure and function of an actual light chain. And so what we're expressing on the surface of the B cell is a pretend IgM molecule, B cell. The IgM molecule, surface IgM is sometimes called the B cell receptor. Okay, we have a pretend B cell receptor, and that will then cause this cell to start to proliferate. Okay, we also at this time, as soon as we make the pretend antibody IgM, we turn on the expression of the Ig alpha beta pairs, which are the equivalent of the T cell CD3 signal transduction guys. Okay, so we've made a heavy chain gene. We're going to amplify the numbers of this, and now we're going to go to work after we've taken that one pressure cell and converted it into the clonal descendants, several hundred clonal descendants. Now we can go to work on each of them, trying to make a functional light chain gene. And many will call if you were chosen kind of thing. So there's a lot of slaughter, but we amplified our numbers, and so some of them are going to eke it on through to make a real IgM, a real heavy chain, and a real light chain. And we first try to make a kappa light chain, and so forth. We're not worrying about that here. But this is just saying that we will try to go ahead and make we've made the heavy chain. Now we're trying to make the light chains here. V and J. This figure doesn't talk about kappa and lambda yet, but we'll succeed that in So these are large pre-B cells because they're proliferating. After the proliferation is over, they become small pre-B cells, and they're doing VJ joining, and now we've made an actual real light chain, and we have IgM on the surface. And that will then, shortly thereafter, we'll start alternate splicing, which will make IgD as well as IgM off of the primary RNA transcript of that gene. Okay. Again, we're going to page 988 here. Pro-Bs are trying to make a heavy chain. DJ and then BBJ. You have several goes at this. Doesn't work, you're out of the picture. You succeed in making a heavy chain, then you're going to have to try to make a light chain. You try first to make a kappa light chain. You work on one of the chromosomes. If that doesn't work, you like the father chromosome. If that doesn't work, you go to the mother chromosome. If that doesn't work, you go to the father chromosome with the lambda chain. If that doesn't work, you go I've lost track of mother and father here, but you go to the mother and try to do the same thing. So you get lots of chances. First father, first kappa, lambda, mother, father, lambda, first kappa, mother, father, lambda, mother, father. So you have pretty good success here when you make a light chain. And all this is, you should know then that isotype exclusion means that you make any B cell that has made an IgM, all the IgM molecules on a surface have the same antigen specificity, of course, but they're only kappa, or for another B cell it might be they're all lambda. You never mind a B cell that's making some kappa and some lambda antibodies on the surface. That's really my isotype exclusion. Don't worry about page 90.